And Ben, you can go ahead and introduce Dr. McGrew.
All right, sounds good. Welcome, everybody. It's my pleasure to be here today to introduce Dr. Alicia McGrew for her seminar. Alicia came to the University of Florida after doing a bachelor's and a master's at Central Michigan University. Her research while at Central Michigan University was looking at the trophic ecology of an invasive crustacean in the Great Lakes. And I thought it was funny that when she came here that, her research was going to go from studying the largest freshwater aquatic ecosystem in the US to the smallest freshwater aquatic ecosystem in the US, the food web that's found in the carnivorous pitcher plant Sarracenia purpurea. And when Alicia came here to UF, I had a couple of ideas of research projects and chapters that I thought would be good for a dissertation. And Alicia listened to them and thought they were really interesting, and then totally came up with her own ideas. And, I haven't been able to, all I've done is just watch her do awesome research, from that day forward, really creative and really independent research. And her journey has taken her far and wide, collecting pitcher plant food webs from Florida to Maine, while driving a minivan with a chest freezer powered by a deep cycle marine battery. It's taken her to the mountains of Switzerland. And all along the way, she's shown just amazing ability to pick up skills, whether it's utilizing particle counting technology that she figured out on her own, post-image processing, statistical analysis and coding, and all types of different skills that have really just made her research exceptional. Alicia has also funded her own research, her research through a research abroad for doctoral students from the University of Florida International Center, her research in Switzerland. And she has really, really excelled as a researcher here during her PhD, as you'll see in a little bit from her presentation that she's about to give. But beyond her research, she's really been extremely dedicated to teaching and outreach during her time here at UF. The three faculty that are in WEC and SNRE that she's TA'd for have told me she's one of the best TAs that they've ever had. One of them has been teaching for over 30 years. And she was honored with a Graduate Assistant Teaching Award at the graduate school level for her work as a TA. But she's gone beyond her TAing, and she's mentored many undergraduate students in the lab, as well as participating in mentoring high school students through the UF Center for Pre-Collegiate Education and Training Student Science Training Program. She's also taken part in the University of Florida Women's Mentoring and Advocacy Program, whose goal is to empower mentees to develop into female leaders during their undergraduate career and beyond. And Alicia also helped organize the Power to Her Women's Leadership Conference. She's also done a lot of outreach within the community here at UF, teaching interactive science lessons on carnivorous plants through K through 12, and judging the Alachua County Public Schools Science Fair. All her outreach and mentoring, she was honored with the Shannon Fitzcoy Memorial Award, which recognizes and supports students who demonstrate a passion for making a difference to youth in their community. And so as you can see, she's been just an amazing student and person here at UF. Personally, it's been a joy to have her in the lab and see her do her research and the influence that she's had over the people that she's interacted with. And, as an advisor, one of my favorite things to do is learn from my graduate students. But Alicia has really taken it to the next level with her dissertation and her research. And I'm going to turn it over to her so she can show you what she's done. So enjoy. Here you go, Alicia.
Thanks so much, Ben. That was really great. Thank you. I'm really excited to be here with you all today to talk to you about some of the research that I did for my dissertation. I actually graduated in August, so I'm currently an alum of UF SNRE, and I just started a postdoc last week. So I'm still, really excited about this research and sharing it with kind of a new group of people. And I'm happy to take any questions at the end that you might have. So most of the research for my dissertation has been focused on this aquatic food web that lives inside the leaves of a carnivorous plant. And I've been really interested in community structure, so the organisms that live inside this habitat. So what is community structure? In studying the organization of ecological communities, we can approach how groups of organisms are structured in time and space through a number of different perspectives or lenses. We might take a taxonomic approach and ask questions about how many kinds of organisms are in a particular community and whether different communities have the same kinds of organisms. We might instead ask how the organisms in a particular community are related to one another and whether different communities are clustered or spread out across a phylogenetic tree or a tree of relatedness. We might also ask how the organisms in the community are interacting in a network, perhaps through trophic interactions in a food web. But patterns of community structure vary pretty substantially across space and time. And unpacking the ecological processes that lead to these patterns is really a fundamental question in ecology. And two of the most well-established controls on patterns of community structure are bottom-up and top-down processes. And bottom-up processes control community structure by regulating the abundance and diversity of organisms at the base of a trophic pyramid, or basically the primary producers. Top-down processes control community structure through predation by limiting the abundance and diversity of prey organisms. And one of the kind of cool ways that predators control patterns of diversity in a community is through keystone predation. So keystone predators are organisms that increase the diversity of their prey species by removing the most abundant organism, or the competitive dominant, like the red squares that we see here. These are kind of like Payne's sea stars when we think about sort of tidal pools. And after that predator selectively removes the most abundant prey item, diversity increases. And we might have rarer species appearing in the community, like these fun green species here. While both bottom-up and top-down processes have really important consequences for all ecosystems, the relative importance of these two processes has been debated pretty heavily among aquatic ecologists. And part of this debate stems from the fact that these processes aren't mutually exclusive controls on community structure, and they can really interact in interesting ways to drive patterns of diversity. And after many, many years of research, the roles of these are still not fully resolved. Because partly, like in addition to these natural processes that we have going on, we have changing bottom-up and top-down controls that result from overharvest of aquatic organisms and eutrophication that can further complicate attempts to disentangle the isolated and interactive effects on community structure. And beyond these differences in bottom-up, top-down processes, there are kind of inherent challenges in working with large aquatic systems. These can include the heterogeneity of physical or chemical conditions in lakes. Lakes are kind of like fingerprints. They're all a little bit different. And the lack of spatial boundaries in marine systems. So this makes it really hard to sort of tightly control explorations of natural systems. And, you know, in the Experimental Lakes Area Project in Canada, where they, had this awesome ability to separate a lake and dump a bunch of nutrients into one side and not into the other, we're not really allowed to do those kinds of things anymore. It would be great if we could. So we have to find a system that works, that we can tightly control, that's aquatic, has some of these similarities of large systems. So to do this, we use the aquatic ecosystem in the leaves of the carnivorous plant, Sarracenia purpurea. They're a discrete aquatic ecosystem that's home to this complex, co-evolved food web. And the model has been used for over 40 years to ask questions about ecological and evolutionary dynamics. And it provides this really ideal combination of a small, replicable, tractable system that has ecological dynamics that are natural and similar to some larger aquatic systems. Unlike other carnivorous plants, Sarracenia purpurea lacks substantial digestive enzymes to capture, to process the prey that it captures and acquire essential nutrients, usually nitrogen. Instead, it houses this really cool, complex, co-evolved aquatic food web that's comprised of bacteria, protozoans, rotifers, and several species of dipteran or fly larvae. In this system, the bacteria are really the powerhouse of the food web, and they're largely responsible for the main ecosystem function of decomposition. And they also serve as the base of the food web and are consumed by two main types of sort of intermediate consumers, or these primary consumers. Those are protozoans and the pitcher plant rotifer, which is considered a co-evolved species. And the top predator in the system is the pitcher plant mosquito larva, Wyomia smithii. So in this aquatic ecosystem, bottom-up processes stem mainly from captured prey, which consists largely of ants in North America, and sometimes a lot of ants. The availability of prey resources has direct bottom-up influence over the decomposers, or the bacteria. So when thinking about bottom-up processes in the system, we're mostly thinking about bacteria. And the availability of resources can alter the taxonomic and phylogenetic composition of the bacterial community, including relative abundances of those bacteria. The top predator, the mosquito larva, directly consumes a lot of the intermediate consumers, but also the bacteria. So the presence or absence or abundance of this top predator can also alter taxonomic phylogenetic composition of the prey in this food web. And it can shape a lot of other different processes through their generalist filter feeding. So in addition to those direct effects, basically trophic interactions, bottom-up and top-down control can have indirect effects on community structure. So changes to the bacterial abundance or diversity resulting from different numbers of prey capture could have impacts on higher trophic levels, cascading impacts on ecosystem function, etc. And changes in the abundance of diversity of organisms resulting from predation might result in a trophic cascade, keystone predation, some of these big concepts that we think about a lot in ecology. So in a trophic cascade, predation by the mosquito larva on some of these intermediate consumers would release bacteria from predation pressure, increase their abundance, and potentially their diversity. And the pitcher plant mosquito has been called a keystone predator in this system for many years. So these indirect effects of preying on a competitive dominant, whether that's a protozoan or a bacteria, have some serious consequences for community structure. So even though this model food web and this ecosystem have been explored for over 40 years, there's still a lot of questions to answer about sort of bottom-up, top-down processes in this system and scaling those up to larger aquatic ecosystems. This is especially true for the microbial community and for patterns and trophic interactions. So with these ideas in mind and this interest in bottom-up and top-down processes, I took several approaches or lenses to community structure and explored three broad research questions for my dissertation. I asked, how do patterns of bottom-up, top-down processes influence taxonomic and phylogenetic community structure of the microbiome or the microbial bacterial community in this system? How do those processes influence size-based community structure, body size-based structure? And in addition to some experimental work, I also did a quantitative literature synthesis looking at whether size-based approaches to community structure are universal. So for my first chapter, I asked two specific questions about the structure of microbial communities in response to bottom-up and top-down processes. The first was, how do they respond? Are nutrients more important? Is predation more important? And secondly, I really wanted to clarify the role of this really important predator in the system, the mosquito larva, and assess whether or not it was truly a keystone predator on bacteria. So to do this, I conducted some experiments, and I did them in the greenhouse in Florida in July, which for anyone who's starting a project, don't recommend, really hot. The regional pools of organisms, so bacteria, I collected in the field in Apalachicola National Forest, which is the closest place where we have natural populations. And I basically put a bunch of pitcher liquid into a vat and homogenized it. But I wanted to remove some of the coarse particulate matter, so captured insect prey, dipteran larvae, etc. And this was the regional pool I used for what's going to be called the BUTD experiment going forward. And for the keystone predation experiment, or KP experiment, I added the second filtration step, where I basically filtered out all of the intermediate consumers to isolate that mosquito larva bacteria trophic interaction. Nobody else is interacting with the food web. It's just that single trophic interaction. So the experimental treatments for bottom-up and top-down control included a 2x3 factorial design of resource level and the number of top predators. And I'll show a little schematic of this. So in the greenhouse, I randomly assigned treatments to a bunch of different pitchers. And each was filled with 10 milliliters of either a coarse filtered regional pool of organisms for the BUTD experiment. And were seeded with their assigned resource and predator levels. So either low resource levels or 5 ants. And these were fire ants collected in Apalachicola National Forest, or high resource levels, 20 ants. And then 3 different levels of predation. So no mosquito larvae, 2 mosquito larvae, or 10. And they're assigned with a treatment code, which is basically a pair of letters representing the combination of resource level and predation levels. So for instance, HL refers to pitchers with high resource levels and low predation pressure. And we'll see this later in some of the plots. And for the keystone predation experiment, I only used 1 resource level, just low resources, and 3 levels of predation. So I allowed these pitcher communities to develop for 2 weeks, which for most of this food web, especially in the summer, is multiple generation time. So a lot of dynamics are happening. And after this, I mixed the pitchers and I subsampled some liquid to send off for sequencing. And the samples were sent to the University of Minnesota Genomics Center for sequencing of 16S ribosomal RNA. And the sequences identified during this process were clustered based on their similarity. And any sequences with more than 97% similarity are considered an operational taxonomic unit, or OTU, which I will use here instead of taxonomic names for bacteria. So I explored patterns of community structure through 2 perspectives here: taxonomic diversity and phylogenetic diversity, or patterns of relatedness. And I looked at patterns both within pitchers, or alpha diversity, and then patterns between pitchers, or beta diversity. So Chow-1 richness is a measure of taxonomic alpha diversity that considers both rare and common species. And I used this abundance weighted metric. And Bray-Curtis dissimilarity is a way to measure compositional differences between communities. And for phylogenetic diversity, I considered 3 different measures of alpha diversity, which I'm not going to talk about right now. And then compositional differences based on relatedness, or phylogenetic beta diversity. So to analyze the response of microbial alpha diversity to bottom-up, top-down processes, I conducted a 2-way analysis of variance, or ANOVA, for the bottom-up, top-down experiment, and a 1-way ANOVA for the keystone predation experiment. And for beta diversity, I visualized compositional differences using non-metric multidimensional scaling, or NMDS, and quantified the role of these processes through a permutational multivariate ANOVAs, or PERM-ANOVA. And then I assessed pairwise differences between resource and predation levels using both dispersion test and pairwise differences in dispersion. So getting into some results, this is the phylogenetic tree of Sarracenia purpurea microbiome. There are over 1,800 known operational taxonomic units. So basically this is matched to some existing sequence that has a name, like E. coli. And these are encompassed by 18 established or candidate phyla. And I think this looks like the Death Star, and I like to say that because it's a fun little thing to insert in there. So first are the results of the taxonomic alpha diversity analyses, where the 6 predation resource predation treatment combinations are on the x-axis and Chow-1 richness is on the y-axis. And in both the bottom-up and keystone predation experiments, Chow-1 richness was higher in pitchers without mosquito larvae, represented here in beige, than it was in pitchers with either low or high levels of predation pressure. But there was no difference between 2 mosquito larvae or 10 mosquito larvae. So basically, to put it simply, we see greater taxonomic diversity of OTUs when predators are absent from the pitcher. And shifting to taxonomic composition of these NMDS plots, the levels of predation here are represented by 3 colors. So none in beige, low in orange, and high in red. So I think red, bloody red predation, just keep that in mind. And then resource levels are distinguished between 2 different point shapes, so circle and triangle. And in both the bottom-up, top-down, and keystone predation experiment, we can see that community composition differs based on the level of predation, with distinct groups of pitchers separating out across this first NMDS axis. So we're basically visualizing distances between communities. So we see the absence of predation in beige, low predation in orange, and high predation in red in the BUTD experiment, and a very similar pattern for the keystone predation experiment. So it appears that there's a clear gradient in community composition from no predation to high predation in both these experiments. And in addition to compositional differences, pitchers in the bottom-up, top-down experiment had greater dispersion or wider spread or dissimilarity between communities relative to pitchers without predation. So these are more tightly clustered and these are spread further apart. And this is just saying that predation is making these communities more distinct or more dissimilar from one another. So pivoting to phylogenetic alpha diversity, I considered 3 different measures of phylogenetic diversity, and they're providing different perspectives on patterns of relatedness in OTUs. So Faith's PD, or Faith's phylogenetic distance, is related to species richness. So PD measures the sum of all the branches in a phylogenetic tree, and this sum was higher in the absence of predation than it was in either low predation or high predation. So instead of seeing this presence-absence effect with Faith's PD, we're seeing a gradient of predation pressure driving phylogenetic distance of bacterial OTUs. And this might be driven by patterns in higher OTU richness in the absence of predation, where higher taxonomic diversity can also lead to higher PD by adding branches as OTUs are added. So MPD, this middle column, or mean pairwise distance, is another measure of phylogenetic diversity. And it's basically the average of the distances or the sum of the branch lengths between every single pair of OTUs on the tree. And MPD is lowest in red when there's high predation. And MPD basically reflects sort of deeper patterns in the phylogeny because it's incorporating deeper nodes. So we're basically looking at loss of entire phylogenetic branches or entire clades. And that's what's happening in this pattern. We only see loss of these branches under high predation pressure. And MNTD, or mean nearest taxon distance, didn't really respond to predation pressure, but there was marginal influence of resource level here, which is the first time that this is cropping up in these analyses, and that pitchers with high resource levels have slightly lower MNTD than those with low resource pitchers, suggesting that some OTUs might be selected for because they can, they're a little bit hardier, they can withstand low resource environments. So in both the Keystone predation experiment, Faith's PD was higher in pitchers without mosquito larvae than with mosquito larvae. So we're back to our presence absence pattern. But unlike the bottom up, top down experiment, resource and predation had no effect on either mean pairwise distance or mean nearest taxon distance. So pitchers without predators include more branches from the phylogenetic tree than pitchers with predators, and some branches are present under low predation and they're absent under high predation. So shifting to compositional differences in these NMDS plots, we're just visualizing dissimilarity between these communities. Predation, again, was important in determining compositional differences in both of these experiments. So phylogenetic community composition is basically measuring the distance, the phylogenetic distance on that tree between one OTU in a community and every other OTU in another community. And this is differing based on the level of predation, so low and high and no predation. So in the bottom up, top down experiment, in addition to some differences based on predation level, there was a significant resource predation interaction, but it's a little unclear here, a little unclear how to unpack it. There's no clear distinct gradient on either the first or the second NMDS axis. And in addition to these compositional differences, in the Keystone predation experiment, there was higher dispersion due to predation. So communities are spread out further apart under predation in either orange or red than they were in beige. So taking a step back from all of these metrics, if we zoom out and just think about this tree as a whole and consider how predation affects taxonomic and phylogenetic diversity from sort of a coarse perspective, the influence on mosquito larvae becomes a little bit clearer. So we see some sort of cosmopolitan phyla, like cyanobacteria that's in every single picture here. And then there's some less common phyla. So the ones in beige are present only when mosquito larvae are absent. So if there are either low or high numbers of mosquito larvae, they're basically eating every single bacterial representative in the community. And in addition, these phyla represented in red are lost under high predation scenarios. So we lose additional branches or phylogenetic lineages under high predation. And these losses are, it would be nice to think that there's some sort of ecological significance, there's something really cool happening. Mostly it's just related to relative abundance. Some of these phyla are just rarer in the pitcher system. So they're getting eaten more easily because they're less abundant. So to sum up the broad impacts of bottom-up, top-down processes on microbial diversity in the system, top-down control is the stronger driver of diversity. It leads to lower taxonomic alpha diversity, lower phylogenetic alpha diversity, or no effect, depending on the metric, and compositionally distinct communities. And one of the aspects of these experiments that I'm most excited about is that we've really demonstrated that the pitcher plant mosquito larva is not a keystone predator. It does not eat a competitive dominant and then increase bacterial diversity. It just eats everything. So it's a generalist filter feeder, and it's leading to lower alpha diversity pretty much across the board. But I think an interesting thing is that many of these communities are very distinct from one another. So is this predator acting as the stochastic force that's driving compositional differences? And how does that play out into ecosystem function? These are some interesting questions for future research. So for this first chapter, we've established that predation is the strongest driver of microbial community structure, but what about the rest of the food web? I wanted to unpack the effect of bottom-up, top-down processes on broader trophic interactions. So I elected to take this size-based approach to characterizing community structure that encompasses both trophic position and interactions. So why do we care about body size? What's the big deal? It's this really cool fundamental trait of every individual, and it has properties that have scaled impacts across levels of biological organization. So for instance, the body size of one protist is related to its metabolic rate, its reproductive rate. And those have consequences for population dynamics, like population abundance and overall growth rate. These scale into, are you big enough to eat certain things or be eaten by certain things? So trophic interactions, which then have consequences for how energy is flowing through the system. So this really simple property that's inherent to all organisms is a great proxy trait for all of these other processes. And one way to conceptualize this is by considering an Eltonian pyramid, where organisms are distributed across trophic levels, and only some energy is transferred from one level to the next through these trophic interactions. But if we sort of shift our perspective on this trophic pyramid, we can intuitively acknowledge that there are a lot of really tiny things. And those are consumed by increasingly large organisms, but these organisms are also increasingly less abundant or decreasingly abundant. So this shift in perspective allows us to consider the transfer of energy through trophic interactions based on distributions of body size instead of taxonomic identity. And one of the most common ways to frame the distribution of abundance or biomass of organisms is by plotting individual body sizes on the x-axis against the abundance of those body sizes on the y-axis. And these distributions effectively remove taxonomic identity. And this distribution is fit with a model that provides information about the shape of the distribution. One of the most, So the most common approach is to fit a statistical model through a log-log plot of these organisms and their abundances. And the models provide two parameters that are really of interest. The first is the y-intercept, which is basically telling us how productive the system is. And this is typically productivity at either the lowest trophic level or the lowest size of a particular group of organisms. And the second parameter is the slope of the line, which is really a measure of how energy is distributed across organisms in an ecosystem. And I kind of think of this as a pipeline of energy transfer that's facilitated through trophic interactions. And these parameters have both been shown to be sensitive to bottom-up and top-down processes in large systems like eutrophication or overharvest of marine organisms. And the most important response is really captured by changes in the slope, which might become steeper here or less steep, depending on what's going on with the distribution of body sizes. So I use these same experiments. Instead, I just pulled the protists out of this instead of the bacteria. So again, these sort of 2x3 factorial combination of resource and predation level, and let the system run for 2 weeks. So one way that body sizes are captured in microscopic organisms is by making a slide and counting things on that slide and measuring them. And that's incredibly time consuming. So I use this really novel and super cool piece of technology called a FlowCam. And it's essentially a combination of a compound microscope, a flow cytometer, and a high-powered camera that characterizes particle properties as they pass through fluid. And I have a video here. I'm just going to skip to like a fun part in the middle, just so you can see what's happening. So this is a pitcher plant liquid sample from one of the experiments. And every single red square is one particle that's being imaged by the FlowCam. And for each of those images, a bunch of different size and shape properties are estimated by the software that comes with this FlowCam. So I can process so many more samples this way than I ever could on a microscope. So using those image galleries, I calculated body size by a property called area-based diameter. And it basically takes every pixel in that image and rearranges it into a circle and said, if you were a circle, how big would your diameter be? And this is sort of a proxy for traits and deals with differently shaped particles. So something like this small protist is very different than this green algae. So it's kind of taking all of those pixels and standardizing for that. And there are a couple predictions we could make about what these size spectra would look like based on resources and predation. We might expect under high resources, perhaps more small organisms because those things have higher turnover time, so they're using resources a little more quickly. We see that reflected in the community. In contrast with predation, we might expect a different distribution where we see that some body sizes are lost. So this happens a lot with overfishing in the ocean, or a decrease in abundance of bigger size classes because they might be more sensitive. And for these experiments, I used two different approaches to map these size spectra and get the slope of the line. The first is sort of a traditional binning method where I chunk all of the organisms up into different size classes, and I calculate the abundance of that size class, and I add one to standardize for any bins that don't have organisms in them. And this is fit with the y equals mx plus b linear regression. And the second approach is a bounded power law and maximum likelihood estimation, which is supposed to be the most robust technique for estimating body size distributions because it doesn't bin individuals. It's basically taking the likelihood that you are bigger or smaller than a particular body size. So in this first method of fitting a size spectrum, I have my 6 pitcher plant treatments with different resource levels, low resources on the top row, high resources on the bottom row, and the 3 different levels of predation in the different columns. And these are binned into 10 micron size classes, and the abundance of organisms in that size class is on the y-axis. So there's some patterns here that stand out in the distribution of points that relate to those predictions about what might happen in these different size classes that I want to just point out. The first is if we zoom in on the smallest particles, we can see that there's some differences in the abundances of the smallest size class across these low resource pitchers, varying between maybe 100 individuals and up to 10,000 individuals. But for high resource pitchers, we consistently see a lot of really tiny organisms. So these differences in abundances of small organisms are probably making the slopes steeper. And if we move up to some intermediate size classes, and this is where a lot of the protozoans are at in these intermediate sizes from 10 to 30 microns, we see patterns both across resources and predation. So there are more individuals in the intermediate size classes under high resource treatments. And then there's also fewer individuals in these intermediate size classes as we move from the absence of predation upwards to high predation. So we're kind of truncating the size range here. We're losing big size classes, which should make our slope also steeper. So instead of looking at a bunch of lines on a plot, let's look at these in a box plot. It's a little more intuitive. So we have our 6 treatments here and the steepness of the slope here. So these are the steepest slopes. And there's a couple cool patterns that come out here. Predation is important, but resources are really important here too. So in terms of resource level in dark green, high resource pitchers have steeper slopes. So the abundance of those really small organisms is making those slopes steeper. But in terms of predation, it's not about the number of predators. It's just the presence or absence of predation. So in the absence of predation here, we find steeper slopes, which isn't quite intuitive because we have more organisms in those intermediate size classes. We might have expected shallower slopes. So this led me to think maybe it's just this method that's not the right way. Like, let's try another method to do this and see what happens. So I use this bounded power law maximum likelihood estimation technique. And in these plots, which are very slowly loading, organisms are not binned. So every individual point here is a single protozoan or rotifer. And the y-axis is the probability that another point is greater than or equal to in size than those other organisms. So it's kind of a proxy for abundance. So if we look at patterns in different size classes of these organisms, we see some similar patterns emerging from the other method. Looking at the smallest sizes, oops, there we go. We can see there's a difference in the smallest size classes as we move from no predation to low predation to high predation in the low resource level. But under high resources, there's pretty consistent number of small particles no matter what. So potentially some resource predation interactions happening here. And if we look at the largest size classes, I think this is where the most interesting thing is happening. There are a lot of really small gray dots in this circle. There's hardly any gray dots here. So basically we're losing entire body sizes. We're truncating the size range. Another thing to point out in these plots is that the dots and the lines aren't matching up very well. They're not really like fitting the data particularly well. So as I'm thinking about this and looking at this pattern and the ANOVA, nothing's really happening. Predation is significant at the factor level, but there's no pairwise differences between levels of predation. So it seems like looking at those dots, there's definitely something happening, but that's not coming out when we look at it in this box plot. So parsing apart the ecological and the methodological factors is kind of important here. So ecologically, predation is consistently decreasing those large size classes. So big ciliate protozoans, rotifers, and resource level is consistently increasing the number of small particles. But that's not playing out in what we see with the slopes, depending on how we construct these size spectra. So the MLE slope estimates and the fitted lines definitely misestimate the abundance of particles. They're not fitting the data particularly well. And this traditional binning method performs better, but you also lose resolution when you bin data. So I'm wondering, what is the deal here? Like, is this because it's small size range of organisms? Is this system just weird? Like, what's going on here? And this kind of led me to my third chapter, which was looking at in the body of literature for size spectra, how do methods influence spectral slope estimates? I had a big problem in my experiment in this like perfect system. How does this happen in an ocean or a lake? How does this play out? And does this make this a reliable tool to use for conservation and management? So to ask this question, I used a meta-analytic approach. And I basically conducted a quantitative literature synthesis of the size spectrum literature. And I asked two questions. What is the influence of methodological approaches on size spectrum slopes? And are there broader ecological patterns in size spectrum that might be obscured by the differences in how these spectra are constructed? And the first particle size spectrum was fit in 1972. So there's a really big body of research to go into asking this question. So I followed a PRISMA meta-analysis procedure to systematically identify, screen, and collect data for qualitative and quantitative analyses. And the primary criteria for eligibility included that it clearly presented a spectral slope, and the methods were either stated in text or I could back calculate the methods from the figures. And if you've ever done meta-analysis or spent a lot of time on Google Scholar, this "please show you're not a robot" might have popped up. So if you're just starting your master's or your PhD, have fun. So as a sort of qualitative overview, the differences in size spectra have been described really generally in some review papers, but this is the first qualitative summary of how spectral slopes are distributed across ecological and methodological factors. So size spectra are constructed in a number of habitat types, but largely in marine systems. And they span a number of organisms from bacteria to benthic macroinvertebrates to fishes. And I considered 4 main methodological factors that have been identified as important in theoretical or empirical size spectrum in the past. And I'm not going to go into the specifics about that. I'm just going to kind of give you the big takeaway. And to sort of get at what was important, I used generalized linear mixed models. Spectral slope was the response variable. Methodological approaches and ecological factors were the fixed effects. And then to account for study-to-study variation, I used a random effective article title. And then I looked at some sort of like post hoc analyses of among organisms, who's different from who. So in this full model, methods really matter. The response variable, or whether you use abundance, biomass, or biovolume, affects the spectral slope estimate, as does this standardization technique called y-axis normalization. Whether or not you used a linear or a nonlinear equation wasn't important at sort of a coarse scale, but that became important later during subsetting, which I'll show you in a moment. And then whether or not you binned and the size of the bin that you use was important for spectral slopes, as was organism type. And bacteria are going to stand out a lot going forward. So to kind of create this framework to deal with these differences in methodology, I created this model subsetting tree. And I ran a bunch of different models by isolating different methodological approaches. And I just want to do 2 patterns. I know I'm like really tight on time. And those are the ecological factors. So across all of these models with different methodological approaches, benthic macroinvertebrate spectral slopes in orange were consistently the shallowest slopes. So there's some sort of cool physiological thing that's leading to spectral slope differences compared to other organisms. And bacteria in gray across models are consistently the steepest slopes. So even though we're constructing these size spectra differently, there's still some cool ecological patterns that are happening. And estuary systems were consistently shallower as compared to lakes, marine systems, or wetlands. And that might have to do with sort of mixing of marine and freshwater systems and productivity there. So the big takeaway here is that methods really matter when you construct size spectra. There's substantial variation in slopes based on these different approaches. And the importance of these different methods is revealed through those subsetting trees, which I didn't really talk about here. But there's also a lot of study-to-study variation captured by that random effect. So the takeaway for this really is stuff needs to be standardized. We need to collect organisms in the same way. We need to be transparent about our methods, have reproducible science. But what I'm so excited about here is we have 50 years of data that no one has been able to use up to this point. So this is kind of a new cool framework to ask broader questions about size spectra, especially with respect to conservation and management practices, and dealing with some of those study-to-study differences in methodological approaches. So some takeaways really quickly. Predation matters for bacteria. predation matters for protozoans in these systems. And this is consistent with larger ecosystems. And when we take these sort of things that we learn from an experimental system and scale them up, we see that methodology really matters. And this is really highlighting the importance of reproducible science. And I'm going to just buzz through this to get to my question slide. Lots of people to thank. Baser Lab members, my committee members, Christine Angelini, Matt Cohen, and Ethan White were awesome during my 5 years at UF. Zach Freeman at Western Virginia, he's at Wisconsin now, who helped unpack the mystery of bacteria for me. Some of my new work family at Northwest Florida State College for helping with FlowCam. And always friends and family are so important for the graduate school process. And with that, I don't know if we have time for questions. If anyone wants to stay on, I'm happy to stay, but otherwise, have a lovely afternoon.
Thank you so much for that excellent presentation, Dr. McGrew. That was awesome. Maybe we can take just 1 or 2 questions. If anybody has any, you're welcome to unmute your mic or to type in the chat. And if you are watching on YouTube, you can also type in the chat there. I have a question.
Sure.
So one of your takeaways was about the importance of kind of standardizing methods. I was wondering if you could talk about like that FlowCam piece of equipment, and if equipment and technology like that could kind of help move in that direction, or what in your mind would help researchers get to a point where they're kind of speaking this language methodologically.
You just cut out a little bit, but I think I got the gist of your question. So I think like the basics of reproducible science are kind of independent from any technology that we use. It's how did we do something? I think sharing data and sharing code now is like really effective at making science reproducible. But in terms of sort of like technical approaches, I think it's important to be very clear about that too. And one of the challenges that we're running into with the FlowCam is that the hardware is really easy to run, but processing all of those images and deciding what's a good picture and a bad picture is a little bit subjective. And there's no published standards for the FlowCam. So this has really been sort of a project with May Giddings, who's at Northwest Florida State College, who's sort of the FlowCam tech there. Her and I like bouncing back and forth, like, what do you think is blurry? Like, what's your quantitative cutoff for blurry? And those kinds of things, and publishing those and talking with other people using this equipment. There's only like 100 in the world, so it's hard to talk to other FlowCam researchers, but that's going to be huge going forward. But beyond that, you know, depending if you're doing wildlife or wetland science, whatever people are doing, social science, just writing very clearly when you publish or you present, and then making code and data available, I think are like the lowest bars for that reproducible science.
Of course, the FlowCam can't solve all your problems, you know.
It's going to change the world. I
Were there any other questions? All right. Well, thank you guys so much for coming, and thank you, Dr. McGrew, for presenting.
Yeah, absolutely. I don't think I put my email in the PowerPoint, but if anyone has questions and wants to email me, please feel free to do so. I'd love to chat with you about carnivorous plants later. They're awesome. All right. Thanks so much, Hannah.
Yep, no problem. And for all the enrolled students, we will see you here next Monday.